We're going to start chapter 7, section 2. This is about the cell structure. This is about all of the parts of the cell. You're going to need to know all of the parts of the cell and what each of them do in a cell. So we're going to talk about our cell, and we're going to use an analogy to learn about our cell and all the parts of our cell. We're going to think about our cell like a city. And when you think about a city, if you want a city to run smoothly and everything to work in a city, everything needs to work together. And in a city, there are various different people, different jobs, different buildings and that make the city function and run and operate smoothly. That's exactly what happens in a cell. Each part of a cell has its own role to play. We're going to compare each of the cell parts to something in a city to help you remember what each of them do. So first, we're going to talk about the cell membrane. The cell membrane is like the city border. The city border is what controls what comes in and out of the cell. It's what surrounds it. That is exactly what our cell membrane does. It is the edge of the cell. It contains everything within the cell, and it helps to regulate what comes in and what exits that cell. It helps protect it from outside forces. It's our border. Your plasma membrane is another word for a cell membrane. It is flexible. It is made up of different parts and pieces. It allows nutrients to enter the cell, and it allows waste to exit the cell. It controls the amount of things entering and leaving a cell. It is flexible. Kind of think of comparing it to like a plastic baggie. It holds stuff inside. It is flexible enough, though, that it moves. It is made up of a bunch of parts and pieces. It is what we call a phospholipid bilayer. If you look at the picture there, you can see that it's a series of what we call a phospholipid. There is a ball, and then there is little tails. The tails are nonpolar. The heads are polar. There are two levels. It's like you took two pieces of bread, smeared peanut butter in between both of them, and smushed them together. There is two outside layers and the inside. It is what we call selectively permeable, meaning that it lets some things move in and out of the cell, but not everything. It's kind of like a control bouncer. It lets certain things in and lets certain things out. Each phospholipid has a polar head, which means that it is attracted to water or hydrophilic, and a nonpolar tail that hangs down, which is hydrophobic. It's always arranged so that the polar heads are closest to the water, and it keeps that nonpolar in the middle, which does not like water. It forms a barrier between the polar and the nonpolar so that substances that will dissolve in water cannot pass through. They're stopped by that nonpolar middle. It is also referred to as what we call a fluid mosaic model, meaning that it is fluid, it floats, it moves, it's not solid and rigid. And it is mosaic. When you think about that art term mosaic means that it's made up of a bunch of different parts and pieces. So the things that float within that fluid mosaic model are proteins, cholesterol, and carbohydrates. So let's talk about what each one of those do. Proteins in the plasma membrane transmit signals to inside and outside of the cell so that it can communicate. It acts as a structure that holds it, gives the cell support and its shape. It also provides a pathway. So if you look at that picture there, you can see kind of looks maybe like a hot dog bun, or I think of it kind of like a straw. It's like a tunnel so that things can move from the outside to the inside of the cell. Some of those actually act as a pump where it will push and squeeze and move stuff through the cell membrane. Cholesterol also is there. If you look, it looks like the little orange dot that is floating in the cell membrane with a little tiny tail of dots down through. Those tails of that cholesterol, that fatty acid, keep those phospholipid tails from sticking together. If you removed cholesterol from your cell membrane, what would happen is all of those tails would stick together, and it would make your cell very rigid, and it would not be flexible and able to move. The little Y-like structures that you see sticking out of the cell membrane there, they're the green color. Those are your carbohydrates. And what they do is they help to identify signals in the environment of the cell. So they send messages to the cell about different things going on inside and outside of the environment. The next part that we're going to talk about is the cytoskeleton. We would compare the cytoskeleton to the steel beams or the structure, the framework of buildings. The cytoskeleton is the internal framework. It's like the internal structure of a building. You know, when we're building skyscrapers, they put up those big metal beams that hold it up and give it their distinctive shape. That's what our cytoskeleton does. Our cytoskeleton is also very important for cell movement and cell division. One of the next chapters we get into, we will talk about cell division, how we create new cells, and that is mitosis. And the cytoskeleton is very important for mitosis to occur. There are different types of your cytoskeleton that are microfilaments and microtubules. Your microtubules are more like an empty straw type structure that has a tube that runs through it. And your microfilaments would be more like kind of like a strand. I think about like, you know, maybe a strand of spaghetti. It's a solid tube that helps connect and provide support. If you look, you can see those, and why I would compare it to a straw versus a strand of spaghetti. Again, this helps anchor things. This helps give this cell support and its shape. And if you look there in my picture, you can see those steel beams that help give a building its support and shape. We would compare the cytoplasm to the lawns and gardens or the grassy areas, like the parks, the stuff that fills in the space in between. The cytoplasm is not exactly liquid, and it's definitely not a solid. Think about like thick gelatin, something that hasn't quite, like a Jell-O who hasn't quite set up, but it's not quite liquid yet either. It encases, cushions, and protects those internal organelles. Remember, organelles are those structures within inside a cell that perform a specific function. The cytoplasm is like a park or your grass where it goes in and it fills in the empty space to cushion everything in between. If you look in those, you can see the areas that are filling in the space around all of those organelles. That is the cytoplasm. The nucleus is what we would compare to the mayor of our town. They are the one that has all of the genetic material. They're the brains. They run it. They're the ones who says what goes on inside the cell. Inside the nucleus, there is the DNA. The DNA is like a blueprint of the city. It's like the mayor is there. He runs it. He's got the blueprints to say exactly how that is supposed to run. And what happens when he needs to send out a copy of the DNA is he uses RNA because he doesn't want to send out his original plan and it get damaged. RNA is a copy of the DNA. The nucleus is surrounded by a membrane called our nuclear envelope. It keeps the chromatin, which is uncoiled DNA, inside. Inside your nucleus, you'll also see in that image down below a dark spot called the nucleolus. The nucleolus is where ribosomes are produced. We'll talk about those in a little bit. And scattered all over the surface of the nuclear envelope are little tiny holes called the nuclear pores. And our nuclear pores are what allow our ribosomes, our proteins, and our RNA to come out of the nucleus. The ribosomes are our manual labor. They're like the workers. They're the plumbers. They're the electricians. There are manufacturers. They're the ones who come together and they make stuff. And what ribosomes make are proteins. They bring together all the raw materials. And they take that copy of the DNA, that RNA, and amino acids, and they take and they make proteins. Proteins are essential to life as we know it, and ribosomes make those proteins. Think of the proteins as the machinery for the cells to function. So those would be like the electricity, the plumbing. For each of these proteins that we have, it has a specific job that makes life possible. The little dots that you see in any cell image are your ribosomes. When we talk about our endoplasmic reticulum, you will see that ribosomes are attached to it. In our endoplasmic reticulum, you will see that we have two. We have a smooth ER and a rough ER. The rough ER has those ribosomes attached to it. The smooth ER does not have any ribosomes. The ER is compared to a highway or a road system. Very much so, because what it does is it moves things along and it sends them through the endoplasmic reticulum out into the cell. The rough ER has those ribosomes attached, and it's going to move those proteins along. The smooth ER is responsible for making lipids, and it helps with things like drug detoxification and metabolism in the cell. If you look, this looks like a series of folds. The part that does not have those little dots on it, that is your smooth ER. The one with the little dots, that is the rough ER. Next comes our Golgi apparatus. Our Golgi apparatus looks like a stack of flattened pancakes. It is like the post office. All of the lipids, all of the proteins, everything that is made there in the endoplasmic reticulum is then sent off into the Golgi apparatus. And what the Golgi apparatus does is it sorts, modifies, packages, and ships it out to where it needs to go into the cell. Whether it is that it needs to be moved out of the cell because there is an excess, whether it needs to be stored, wherever it needs to go, it's the Golgi apparatus's job to send it where it needs to go. And if you look, that looks like a stack of pancakes that somebody like kind of tried to pick up, and it's kind of drooping over and syrup is falling off of it. Lysosomes are found in animal cells. They are like the recycling plants. They break down the big macromolecules, and they break it down into something that can be used, or they just completely digest it if it is taking up too much room. It keeps too much extra stuff, or that big bulky stuff, from building up in the cell. And basically, I kind of think they look kind of like a gumball. They're just a little round ball-like sac or structure in the cell. Vacuoles, they can be found in both plant and animal cells, but they are much smaller in a plant cell than they are in an animal cell. And what they are is they're like a water tower, or they're like a warehouse. They store all of the extra stuff. So if you look, this is from a plant cell. You can see those big vacuoles. They're just round ball-like structures that store excess material. The mitochondria is like the energy plant. This is where cellular respiration takes place. Energy is converted from the glucose into the usable currency of the cell, which is ATP, which stands for adenosine triphosphate. Mitochondria actually have their own DNA. You get your mitochondrial DNA from your mother. It is passed down from mother to mother to mother to daughter, and yours is the exact same as your mother's, which is the exact same as her mother's, and on and on. It changes very, very little over time. It doesn't matter whether you're male or female, your mitochondrial DNA comes from your mother. And we also think that this was a prokaryote that moved inside another prokaryote. They evolved together. So it is a very primitive form of DNA. I always think these look like a bean, kind of like with a folded up structure. That folded up structure is where cellular respiration occurs, which means that's where glucose is converted into a usable energy. There are 3 structures that are different in plant cells, and they are not found in animal cells. They are cell walls, chloroplast, and a central vacuole. The cell wall, I like